Welcome to a new global IMMUNOTALK. It is my absolute pleasure to introduce today our global immuno speaker, Professor Judith Allen. Judith is an internationally recognized scientist for her contributions to the advancement of immunoparasitology. She is currently a professor of immunobiology at the University of Manchester in the UK. Now, Judy had a very interesting career path. She first obtained her bachelor's from Bates College, and between her bachelor's and master's and PhD, she spent quite a few years in industry. Quite some time at Cetus Corporation, where she developed diagnostic kits for chlamydia. I think that got her interest in part in chlamydia, and then she decided to start her PhD at the UC Berkeley. And during this time, she identified antigenic peptides in Chlamydia trachomatis. Now, after that, she really followed her passion about trying to start understanding the immune responses to nematodes and moved to London and joined Rick Masel's lab for her postdoc. And subsequently, she stayed around there. She moved to Edinburgh and was at the University of Edinburgh for quite some time, where she established her own independent lab. And since 2016, she has been, as I said, at the University of Manchester. It is there where Judy has continued to study the immune response to metazoan parasites. Her laboratory has made textbook contributions on the response of macrophages in the context of these macroparasite infections. And also she's very interested on what are the responses of the host when they're challenged by multiple, micro and macro parasites. Because of her outstanding work, Judy has received numerous recognitions. She has received the highly cited researcher recognition in multiple occasions, and she's also a fellow of the Royal Society of Edinburgh, the Royal Society of Biology, and the Royal Academy of Medical Science. Now, on a more personal note, Judy and I once published a paper back-to-back. And, this can be very exciting, but also could be stressful, and uncertain times. And I want to acknowledge that I really valued a lot our very open communication during that time, something that is a very valuable experience that we have as scientists. So I'm sure we are in for a treat with Judith's global immunotalk entitled Type 2 Immunity, Learning from Helminths. Thank you, Judy, for accepting our invitation.
Thank you so much. Yeah.
Thank you. So, as we all know, we love to learn more about our speakers. So we have a question for you, Judy, today. You have had a long-lasting career in immunoparasitology, and we would love to know what has been, in your experience, the most transformative advances that have influenced your research.
Thanks, Carla. Well, obviously the type 2 immune field has just, blossomed in the past many years, and there's a lot. But if I start really sort of at the beginning, when I started my PhD, I was actually, even though it was UC Berkeley, I was physically located at UCSF. And just then in that previous year, Mossman and Kaufman had described that T cells could be subdivided into T helper 1 and T helper 2 cells. I wasn't even until I thought about this, realized the timing of that was really quite amazing for me. And although I was in a pure sort of microbiology chlamydia lab, I was actually learning my immunology in Rich Laxley's lab a few floors up, where they were really studying this in Leishmania. And I think one of the important things is the finding in and of itself wasn't so amazing that T cells could produce these different cytokine profiles. But what it did, along with the labs, several labs that were sort of studying it in Leishmania, was it sort of created, I think, a light bulb moment for people sort of understanding that dichotomy of disease presentation. That there were individuals who were protected from intracellular pathogen infection that would manifest with these pro-inflammatory responses, and then the people who manifested with a much more what we now know as a more type 2 response were susceptible. And then, of course, subsequently, people here, Catherine Elson, Richard Grensis here, were able to show that that type 2 response was so critical for getting rid of gastrointestinal nematodes. And of course, I became, completely captivated by that dichotomy. And I think you'll hear that today. That's still, really amazing to me that we've evolved these two sort of, well, more than two, but really essentially two fundamentally different pathways. And then, the work that Maria Yazdanbash and Rick Maisels did on seeing that helminths, not only induce Th2 responses, but these really profound Treg responses. And it just goes on and on. And of course, with my macrophage work, the list goes on, the work from Florent Guinhou and Frederick on origins of macrophage. So I've had many, many influences, but I think one of the things that I think about all of them is that it's not so much the discovery in itself. And I really think the Mossman-Kaufman discovery is a good example. The finding itself is not that amazing. It's that it suddenly opens a window. Suddenly there's clarity that you didn't have before. And of course, then that opens a whole new door for, different things. But so, yeah, there have obviously been many, and there are those, they really are just light bulb minutes when you suddenly see why something is happening the way it does. Yeah.
That's wonderful. And how nice to have the experience of having lived that time, but also now go back and being able to see how impactful it has been in advancing our fields. Yeah. Judy, thank you so much. We very much look forward to your talk. Let's see if you can share your screen.
Okay. Oh dear. Sorry. Just
take your time.
Yep. As always, these things don't happen the way you think they're going to. Okay. So here we go. Share. I'm on a different. This was going to be easy, right? And then it advanced portion of screen, share. There we go. Okay. So is that clear?
Yes, but you want to go to your first one, right? I think you're in your second one.
Yeah, great. Thank you. Thanks so much. And yeah, thank you, Carla and Alina. This is just, as you know, and I'm sure everybody tells you, an amazing series. And it's just really an honor to be part of it. So thanks so much. Yes, and I think, as I've already said, that I'm fascinated by helminths and the type 2 immune response. And I think we've learned a lot from it, not just from the helminths, not just about them, but about our own immune response more generally. And this is a slide that I frequently use. I'm very oversimplified, but as I said earlier, I'm very struck by how completely different the type 1 response is from the type 2 response. And including in the type 1 response, although we know this is more complicated than that, both Th1 and IL-17 responses as they're pro-inflammatory antimicrobial responses. But the responses that we mount to helminths are really fundamentally different. And I'm going to focus on sort of the core cytokines, IL-4 and IL-13, that both signal through the IL-4 receptor alpha. And as I mentioned earlier, I mean, we know that this response is important for helminth infection. And really from the sort of seminal work, much of it done here in Manchester, that these cytokines drive mucus production from epithelial cells, drive epithelial cell turnover that help us get rid of gastrointestinal, these large multicellular pathogens from our gastrointestinal tract. But I think as many people know, my interest really came in understanding what the role of IL-4 and IL-13 was in terms of macrophages, because we began to note that during helminth infections, and this was when I was working as a postdoc in Rick Maisels' lab, that macrophages take on a very distinct, well, first of all, that macrophages were there. I mean, when we started this work, I think people didn't really think about type 2 immunity having a strong macrophage association, but in fact, they're there at very high numbers in almost any helminth infection. And my interest was very much specifically in parasites that are helminth infections where the parasite lives in the tissues, not the ones that are necessarily expelled from the gastrointestinal tract. And what we found, and sort of listing some of the people in my lab that were involved in this work, was that they took on a very distinct transcriptional profile with the very abundant proteins that are now well known in the type 2 community, and many groups, including our own, discovered that these macrophages make very abundant level of these secreted proteins, YM1, realm alpha, and arginase. And you can see here now in an IL-4 receptor knockout mouse during a helminth infection that these proteins are IL-4 receptor dependent. And so the question that has dominated my lab for a long time is, how are these nematodes that live in the tissues, how are they killed? I mean, we know that a gastrointestinal worm is expelled through mucus production, et cetera. But how are these worms that live in the tissues? And these macrophages that are there in such abundance, are they involved in killing worms? And so what I'm going to tell you right now is I'm not going to answer those questions. In fact, I don't have the answer to those questions yet. I've managed to get a lot of funding over the years trying to answer them, and instead have found interesting things along the way. And in fact, I'm really not going to talk about macrophages much at all. And that's because I'm giving the myeloid network talk in a couple of months. But I would encourage people who are interested in that side of things to come. Connor Finley, a postdoc in the lab who's recently left, has found that there's fundamental differences in myeloid dynamics between different strains of mice that really sort of challenge some of our basic understanding of these cells. And it's also a lot on filarial nematode infection. And that really addresses much more the sort of anti-helminth part of those macrophages, but I'm going to focus really on the molecules that they produce, YM1 and realm alpha in particular. Because our idea very originally was that if we could understand what these proteins that these macrophages are making were doing, we could really understand what the function of these macrophages were. And what we found very early on was that these macrophages were strongly associated with physical injury. And as time went on, as we did much more intensive transcriptional analysis, it became clear that it wasn't just these genes, that many genes associated with tissue repair and matrix remodeling were highly expressed in response to IL-4 and IL-13. And then, of course, work from Tom Wynn in particular and others, we now know that IL-13 is one of the most pro-fibrotic or pro-scarring cytokines after TGF-beta. So you have this response that's highly induced by IL-4 receptor signaling and IL-4 and IL-13 that's involved in injury and repair. And so why is that? And I think, it's now pretty obvious, but at the time it wasn't that obvious to me. But these large metazoan parasites that migrate through our bodies are and can be very physically damaging. And I show here as an illustration the hookworm parasites, which I'm going to be talking about the Nipistrongelus system in a while, not a hookworm per se, but has a very similar life cycle. And just to go through it, both to illustrate the level of physical damage that these parasites do, but also just because the life cycle is important for subsequent parts of the talk. But what we know is that the parasite comes in through the skin, through, say, a blade of grass. In fact, I grew up where hookworm was endemic and therefore was never allowed to go barefoot as a child because the parasite comes up through a blade of grass, it burrows through the skin, it then gets into the bloodstream and it gets swept from the bloodstream to the lung. It breaks out of the lung vasculature into the lung, causing a fair amount of bleeding. And then it matures, it molts, it crawls into the airways and up the trachea, you cough, you swallow it, and it ends up in the gastrointestinal tract, where in the case of hookworm parasites, it actually sucks blood from the gut wall. And that's also a very damaging process. So these are parasites where they are causing a great deal of simply physical damage that the immune system has to handle. And what I think that we've become increasingly aware of is that this process of tissue repair is integrally tied to our ability to control these parasites. So not only do we need to repair the damage that they do, we need to keep the numbers of parasites down for our own fitness. And I think during the course of evolution, the body saw these as physical injury. It used these pathways to control the damage, and those same pathways then got co-opted essentially into controlling parasite numbers. And a really brilliant example of that is arginase, a molecule that's well known to be involved in tissue repair and fibrosis, but Nicola Harris's group has shown that the actual downstream products are directly toxic to nematodes. So these processes are really integrally tied together. So because we had discovered, as I say, along with others, these molecules, YM1 and realm alpha, we set out to ask whether they are also fall into this category and whether they're involved in tissue repair. So Tara Sutherland, who was a postdoc in the lab, is now an independent PI, was asking the question about whether YM1 can contribute to repair. And so what is YM1? So YM1 is a a member of a family of proteins that are called chitinase-like proteins. And the parental molecules are acidic mammalian chitinase and chitotriosidase. These are active chitinases. They break down the chitin that you and I are breathing in all the time from insect debris, fungal material. So chitin, of course, is a sugar that needs to be broken down because it can be very large. But acidic mammalian chitinase and chitotriosidase have undergone extensive gene duplication. And these products, these chitinase-like proteins, are very similar structurally to the active chitinases, but they have loss of function and have lost the ability to cleave chitin. They can still bind it, some of them still don't even bind chitin anymore, but they're all lectins, they all bind sugars. And they're very, very strongly associated with chronic inflammatory conditions. So the other thing that to me is incredibly fascinating, and we don't really have the answers to why this is that all mammals have a different set. So all mammals have gone through this gene duplication, actually even plants. And mice have YM1, YM2, and BRP39, whereas humans have YKL40 and YKL39. But why do we really care about this is that in people, YKL40 is really strongly associated with poor prognosis in a huge range of diseases, and yet we really don't know what this molecule does. So it correlates with reduced lung function in asthma and COPD. There's multiple GWAS studies that have identified the gene as a candidate gene for asthma susceptibility. It's associated with poor prognosis in many types of cancers. If you were to type into PubMed YKL40 in cancer, you'll get hundreds of papers. And it's also a biomarker for disease severity and poor prognosis in a range of fibrotic diseases. So this molecule is bad news, and we don't really know why. And so during the first few months of lockdown, Alistair Chenery, a postdoc in my lab, helped out the group that were doing, Tracy Hussle and John Granger, who were doing an amazing analysis of Manchester patients with COVID-19. And we just looked at YKL40 in the serum. I have to say, we did this in part for fun because the outcome to me was so predictable that we would see this. And indeed, YKL40 levels in the serum correlate with disease severity. And we've never published this, but other people now have. So it's just an illustration in a timely way of how this molecule is just so associated with disease, almost any disease. So we really believe that YM1, although not the direct ortholog, is a great model for YKL40. I won't go into all the reasons, but they have similar expression profiles, similar structurally, many, many different similarities. And so one of the reasons we study YM1 is to get a grip on understanding of what YKL40 might be doing in humans. And we've been starting to work very closely with Tony Day here in Manchester, who's an expert on glycosaminoglycans. And we know that these molecules bind sugars, they bind glycosaminoglycans, and understanding what their actual substrates are is going to obviously be an important part of figuring out what they do. But these molecules are structurally very similar, and they're found anywhere and everywhere in the body. So in trying to figure out whether YM1 is involved in repairing tissue, we turn to everybody's favorite model in the type 2 field, Nipostrongylus brasiliensis, that has this lung migration stage that I was talking about, in which you have damage to the lung. And of course, Bill Gauz's group had shown that you need type 2 immunity, and particularly these alternatively activated or IL-4 activated macrophages to repair this damage to the lung. So we thought it was a good model to look at what YM1 might be doing. And this is what I'm going to be describing for the next few slides. Is really all work that Tara Sutherland did. And what we did is when we looked at Nipostrongylus, we saw that in the early time points that there is innate, and many people who work on lung know this, you have YM1 in the alveolar macrophages, and there's a little bit of realm alpha around, and that's all IL-4 receptor independent in the first couple of days of infection. And I should illustrate what happens here is the larvae are in and out of the lung within 48 hours. And then you have this massive lung injury that's associated with the migrating worms and a massive neutrophilia. And the neutrophilia is actually critical for the lung damage, something that once again, Bill Gauss's lab showed very nicely, and we've seen it as well. So you need that incoming neutrophilia to drive the damage. And then the neutrophils subside, the lung injury subsides, coincidental with the rise of the type 2 immune response. And of course, now we know that this resolution actually requires the type 2 response to come on board. And as the type 2 response gets very large, and Nippus strongylosus is a sort of a fantastic model for a sledgehammer of type 2 immunity in the lung, you begin to get very large amounts of YM1 and REL alpha that now are largely IL-4 receptor dependent. And this is just a picture of an uninfected lung. You can see, I hopefully can see some red in the alveolar macrophages. And then once you get infection, you get increase in the amount of YM1, but now you get REL alpha in the epithelial cells. So although originally described in macrophages, REL alpha is, I think, especially in the lung, typically present in the epithelial cells of the airways. And so what Tara did was to use a monoclonal antibody to YM1 to block this early innate YM1, giving them antibody at day -1, 0, and +1 to see what happened if you got rid of YM1. And to summarize data that's long since published, what she found was that YM1 induces a population, an expansion and an increase in the gamma delta IL-17 producing population. This is via the IL-1 receptor. And that turns on the neutrophilia. And you then get the neutrophils that cause this enhanced lung injury, but those neutrophils also form a trap around, well, not neutrophils trap per se, but they coalesce around the worms and compromise worm fitness. And so early on in infection, that early release of YM1 is a trade-off between sort of reducing the fitness of the worms, but also causing lung injury. And so that was great and very interesting. But another thing that was a bit surprising was that she found that when she looked at this subsequent type 2 immune response, when she blocked that YM1 early, this subsequent immune response was highly muted. So this is just showing IL-13, but it was true of other parameters of the type 2 immune response. So if she blocked YM1, she also got a failure to fully mount the type 2 immune response. And when we now knew at this point that YM1 was inducing IL-17, and so we looked at the IL-17A deficient mouse and saw exactly the same thing, that in the absence of IL-17, we also failed to get this type 2 immune response. And that was quite unexpected. That we really had not seen this before, this idea that you would need 17 to mount a type 2 response. And I would say, I think you'll see this later on in the data as well, that YM1 often mimics IL-17. So we think a lot of the actions of YM1 are likely due to its ability to induce 17. So what we were able to see here is that we had this early, very innate response where YM1 is driving gamma delta IL-17 cells, but then they are needed, or the IL-17 anyway, is needed for this subsequent type 2 immune response. And now much larger amounts of YM1, IL-13 driving mucus production, all the other things associated with type 2 immunity in the lung. So about 5 years ago, or a little more than 5 years ago now, my lab moved from Edinburgh to Manchester. And one of the first new postdocs that came on board was Yasuta Sahendra. And this is actually his, well, next week is his last week in the lab, which is very, very sad for all of us. This picture was taken about 5 years ago, and I looked at it and had to laugh because I think I've aged him. He looks a lot older 5 years on. But the question that he was given for this was, why do IL-17 deficient mice fail to mount a full Th2 response? And he's just done a brilliant job over the past 5 years to really begin to answer this question. So one of the first things we did was to look at the transcriptomics in the lung of the IL-17A deficient mice infected with Nippus strongylosus. And when we looked at that, what we saw is the pathway that was the most downregulated or upregulated, I'm sorry, in the IL-17 knockout mouse was the interferon gamma pathway, looking at pathway analysis. And I just want to say that for all the sort of transcriptomic and a lot of work in that my lab does, an amazing research assistant in the lab who's now a PhD student in the lab as well, James, does this analysis. And although that's a very important role, one of his more important roles is to order dim sum at our local Chinese restaurant. But he's a critical player in all the stories that I'll talk about today. So we had this situation in which blocking YM1 or IL-17 deficiency both led to a defect in type 2 immunity. They also, of course, led to a defect in neutrophils. So Jasutis got rid of the neutrophils and found that that was not responsible for the impaired type 2 immune response. So it isn't just the physical damage in the neutrophils that's leading to the type 2 immune response. And one of the things that we had observed consistently over the years in different models, most of it not published, is that YM1 suppresses interferon gamma. And so we'd seen this transcriptional analysis that James had done that now suggested that, that 17 was also suppressing interferon gamma. So the hypothesis was that maybe YM1 is inducing IL-17, which we'd seen, and that maybe it's the IL-17 that's suppressing the gamma. So Jasutis set out to test that. And the first thing we just had to see is there a suppression of interferon gamma? And there is. 2 days post-infection, basically all the gamma-producing cells are producing less gamma in Nippus strongylis. And then when we look at the IL-17 deficient mouse, that gamma is recovered. So it really was showing us that there was that it was IL-17 that was responsible for this suppression of interferon gamma. And the other thing that was quite interesting and important, I think, is that it seems to be restricted to the lung. So if we look at our Nippus strongylis infection, then this ability of IL-17 deficiency, well, the ability of IL-17 to drive TH2 or the failure of a TH2 response in an IL-17 deficient mouse was happening only in the lung. The TH2 response in the gastrointestinal tract was completely normal. So in collaboration with Richard Grensis's lab with Stefano, Jasutis looked in Tricuris. We were just kind of interested to look at a model where there was no lung migration stage to see what the role of IL-17 was. And like we had seen in Nippus strongylis, there was no defect in the gastrointestinal tract. But somewhat to our surprise, there was a major defect in the lung. So it turns out that in Tricuris, even though there's no lung migration stage, that you do get a type 2 immune response in the lung. And if you don't have 17, you don't get that full type 2 response. So it isn't just a matter of mucosally associated T cells circulating through from the gut to the lung. You actually need some sort of IL-17 insult in the lung to get that type 2 response in the lung, and how or why that's happening in Tricuris, we don't know. So at this stage, this is where the model was at. We have, as I say, YM1 inducing 17, and then 17 is suppressing gamma. And then this leads to a type 2 immune response. And I'm not showing the data, but Jasutis was able to confirm that this is actually what was going on by blocking interferon gamma in the IL-17 deficient animals. So we know that this is what's happening here, that IL-17 is suppressing gamma, and that's leading to the development of the type 2 immune response, which now then initiates the whole repair response. So I think I have the same slide twice. I'm not sure why that is. So we have this situation in which we know that a lot about what's going on now with this early innate YM1 response, that it enhances tissue damage and worm killing, and it promotes type 2 immunity via IL-17. So the next thing that really wanted to know is, now when you have this injury response and you have the TH2 response fully mounted and you now have much more YM1, what is YM1 doing there in this adaptive stage of the response? And just for the sort of chitinase-like protein aficionados, I wanted to point out that we also study YM2, that YM1 and YM2 are very similar molecules, but people don't really look at them carefully because they can be quite hard to distinguish. And what's clear is there's virtually no YM2 in a naive animal. So YM2 only comes up during actual nematode infection or helminth infection. And when we look at that, the YM1 is in the macrophages, as it's been described before, but YM2, like realm alpha, is essentially epithelial cell derived. So Tara blocked YM2 at later stages of infection and asked what happened. And this slide summarizes a huge amount of data that I think much of it is quite remarkable, is that, so first of all, what we saw is that when she blocked YM1, she blocked, or delayed, blocking is way too strong a term, she delayed lung repair. But what was more remarkable is that she took recombinant YM1 and gave it to an IL-4 receptor deficient animal that doesn't repair its lung well. She could now drive repair with recombinant YM1 alone. And the other thing that was a big surprise for us was that YM1 appears to drive realm alpha. So we'd always found them associated, but we didn't know that one was actually directly linked to the other. And this is just shown here, is if she blocks YM1 using the anti-YM1 treatment. So here's a control treated animal, here's an anti-YM1 treated animal, you lose that epithelial cell realm alpha. And we know, as other people have shown, and as we show in this paper, that realm alpha is an important regulator of lung repair. So it looks like, so first of all, you know, the big question, is YM1 involved in repair? The answer, as you get into adaptive Th2 driven responses, yes, YM1 is important in driving repair. Mechanistically, how was this happening? Well, we had a big clue from work that of Sabine Emming's lab in Cologne. So she had shown, and Tara and I had been in a small way involved in this study. She'd shown in the skin that IL-4 receptor signaling to macrophages was critical for the rate and of repair, and that this involved realm alpha. And this pathway that I'm showing here is what she showed in the skin, and Tara was able to replicate in the lung, is that YM1 acts on epithelial cells to produce realm alpha. Realm alpha then acts on fibroblasts to drive the production of a gene called PLOD2, which encodes for lysyl hydroxylase 2. Now, lysyl hydroxylase 2 is an enzyme that cross-links collagen in a particular way, giving the tissue mechanical sort of stability. Now, you wouldn't normally find lysyl hydroxylase 2 in the lung or in the skin. It's in places like cartilage, but it gives the tissue sort of strength and I say mechanical stability, but also can lead to scar tissue. So it looks like YM1 induces realm alpha, and these things, which are initiated via the IL-4 receptor, are involved in the sort of rapid restoration of tissue integrity, but also might lead to scar formation. And I think this fits the whole association of type 2 immunity with scar tissue. And I'm going to come back to another point. Well, 2 points I want to make is that this is just really the tip of the iceberg. The reviewers of the paper really wanted us to show that everything YM1 was doing was via realm alpha. Well, I doubt A, it was a hard experiment to do, and B, I doubt very much that's the case. I think this is just going to be one of the many, many pathways in which YM1 acts, is to regulate collagen via realm alpha. And so I think this is just a wide open area to understand more about how type 2 immunity mediates repair. And of course, we already know that IL-13 does other things that are related to repair, like producing mucus and other aspects that are important in tissue balance. So we now were at, back to this slide, we now have this situation in which we now that distinctly from what it does during the innate stages where YM1 helps promote damage and promote type 2 immunity, now YM1 is directly repairing the tissue damage, likely via, at least in part, via realm alpha. But when Tara blocked YM1 at this adaptive stage, she also saw something else quite interesting. She saw that she had an enhanced type 2 immune response. So it looks like YM1 completely opposite than what we saw during the innate stages when she blocked YM1 at day -1, 0, and 1, where we didn't get the full development of type 2 immunity. Now, when she's blocking it as the type 2 immune response is becoming established, now we get an enhanced type 2 response, suggesting that YM1 is actually suppressing the established response. And once again, of course, we're thinking about IL-17 a lot, and Yesutis was doing a lot of studies trying to understand the role of 17. And so he blocked IL-17. So this is distinct from the experiments where he's using IL-17 knockouts, which have no 17 from the beginning. Here he's blocking out IL-17 after the response has already initiated. And he sees just the same thing, that when you block 17, you get an enhancement of the type 2 immune response. So showing that both YM1 and 17 are involved in controlling the type 2 immune response. So, so we now have this situation where we have IL-17 and YM1 blocking interferon gamma that licenses and permits the production of the type 2 immune response. Of course, we know from work that that Rich Loxley's group has done, is that this type 2 response is critical for shutting off this early 17 surge. So we know that this is an important circuit. So we've now found that 17 is driving the type 2 response, but once the type 2 response is established, then you get the suppression of this early 17 response. But now this new data is saying that in addition, once the type 2 response becomes established, whatever residual IL-17 producing cells that are there, they're putting a cap on this type 2 response. So you're not getting too big a response, too large a response. And of course, YM1 is involved in that. And what we don't know is how YM1 is doing that. So YM1 could be putting this cap on two different ways. One is we know that YM1, as I've said before, has the capacity to promote IL-17. But we also know from work that Mira Nair did, and when she was in David Artis' lab and subsequently as well, is that realm alpha can also suppress type 2 immunity. So YM1 may be independently suppressing type 2 immunity, but also Mira has shown in very different contexts that realm alpha itself can initiate 17 responses. So we have a lot of questions here, but I think it's clear that YM1 and IL-17 in some way are putting a lid on this response, and so type 2 immunity is containing that early 17 response from getting too big, and then once the type 2 response is established, IL-17 is containing that type 2 response. So we now have this complete sort of dichotomy between what happens during the innate stage and what happens during the adaptive stage. So innate YM1 is enhancing tissue damage and promoting type 2 immunity, and then it is involved subsequently in repairing the tissue damage and limiting the type 2 immunity via either IL-17 or realm alpha or both. So all of this is in the Dendrostrongylus model, although we've been confirming it in some other models, as I sort of indicated. So I think that this is really something that needs to think about, this relationship between type 2 immunity and IL-17. We know that, and I like this drawing, although oversimplification, but I like to think about it this way, that Alvaro Diaz, a good friend, drew for a review we wrote a number of years ago, is that at this type 2, type 17 axis, there's a lot of pathology, and there's a lot of data out there on things like severe asthma, helminth pathology, fungal pathology that fall in this space. Of course, we know type 17 also plays a big role in this axis, autoimmunity. And importantly, the chitinase-like proteins hang out in this space associated with this kind of nastiness response. So really the question, and I'll just be leaving this question out there, is when chitinase-like proteins, IL-17 and type 2 immunity are associated with these severe pathology outcome, has this counter-regulation just gone wrong? So this regulation that we're talking about here, in which 17 in which type 2 immunity limits 17 and which 17 limits type 2 immunity, is that circuit failing when we have severe pathology? And I've been talking a lot about IL-4 and IL-13 sort of as one, because we use the IL-4 receptor knockout mice a lot. And that doesn't distinguish which one we're looking at, but an obvious interest in the lab really has been whether it is actually IL-4 or IL-13. And two postdocs in the lab, Alistair and Sylvia, along with James's help and Tara's help, as well as Carl Cadler, who's one of our matrix biologists, wanted to ask the question about all this stuff that we're seeing in the lung. Is it IL-13 driven or is it IL-4 driven? So we used IL-13 knockouts to ask that question. And I should say, importantly, these mice had absolutely no defect in IL-4. So IL-4 levels were completely normal. And what we saw was that indeed all our favorite molecules, realm alpha, YM1, AMCAs, they were heavily missing or downregulated in the IL-13 deficient animals. So it looks like these animals are highly deficient, and that most of these molecules are at least largely driven by IL-13 in the lung during nipostrongelous infection. And this just shows that epithelial cell realm alpha is in the wild type animals and how diminished it is in an IL-13 deficient animal. But what's interesting and important, but not surprising, is that the acute lung injury that we see during the nipostrongelous model when the parasite migrates through the lung is much more severe in an IL-13 deficient animal. So IL-13 is really important in controlling the damage. We actually think IL-13 has a big role to play in controlling bleeding. The bleeding is much worse in the IL-13 deficient animals. So what we find is that IL-13 is host protective during this acute lung injury model. And I'm not sure how good a model it is for nematode infection per se, but I think it's a really good model for a type 2 mediated acute lung injury. And IL-13 is host protective. But for the last 10 minutes or so of the talk, I'm going to talk about a situation in which IL-13 in the lung is actually quite bad news, and that's during COVID-19. And my group, like many others, spent some of our life in the past couple of years working on COVID-19. And what we see there is really a surprising story, I think, on how damaging IL-13 is in COVID-19. So this is a story, this is an amazing collaboration. This is a story that started at the University of Virginia, well, and continues at the University of Virginia, when Bill Petrie contacted me to say that they were seeing evidence of IL-13 in their being a player in their COVID-19 cohorts. And this work, I was being led by a PhD student in his lab, Ali Donlin, and Ali and Bill and I had already been talking quite a bit about the role of IL-13 in C. difficile. So I think it was kind of natural to start talking about it here. So they contacted us, and what they had seen was that in their cohort of patients, they were seeing enhanced IL-13 in their University of Virginia COVID-19 patients, and that the risk of ventilation was higher with your higher levels of IL-13. And then they replicated this or validated this in a cohort from a different institution from the Virginia Commonwealth University Medical Center. So they were seeing this potential role for IL-13 in COVID-19. And what started then was one of the most amazing and fun and satisfying collaborations of my career with Bill Petrie and his gang. And we literally had, for the past, until quite recently, and I think we're starting up again as we think about this, for almost a year and a half, we had weekly Zoom meetings. One of the sort of benefits of COVID is how we've learned to collaborate across the miles. And Tara was a central player to this. I didn't take pictures in every Zoom, so I didn't have everybody, but and I won't go through everybody's names, but these are the people that have been involved in the study. And Tony Day here in Manchester has been really helped us understand part of this study, and I think you'll see why in a minute. So what they had done, and most of this starts in Virginia, is that they got the ACE2 mice that can be infected with SARS-CoV-2, and they looked at their immune response in these mice. So importantly, these mice get very severely ill, and they die within about a week of the infection. And they saw that indeed these mice were mounting a type 2 immune response, nothing like what you would see in Epistrongylus, but definitely a detectable increase in YM1, realm alpha. And then when we looked at the sections here, we could see that the type 2 response was there. And then what they did, Ali with Barbara Mann, they began to block IL-13 in these mice. And the results I think were quite remarkable. I'm not sure the figure shows it as well, but Barbara and Ali said how it was just visually obvious which mice had been treated with IL-13, with antibody to IL-13. So if the IL-13 was blocked, they had a much better clinical score, and they were much healthier, and they survived for longer. So blocking IL-13 was having a protective effect in COVID-19, in the mouse model of COVID-19. And importantly, there was no difference in viral burden. What was really fun is that Bill got talking to somebody, and this is once again not my part of the story, and I'm sharing, I should want to make it very clear how much of this was done in Virginia, but they got information from the online Trinetics database, which has, hundreds of thousands, well, millions of patients on it, and who have been treated with biologics. And they found that the 81 patients that had been treated with dupilumab were not in their cohort who ever had severe COVID. And so when they took the data and looked more closely and matched 65 of the patients that they could match for comorbidities, all the things you match for in this kind of study, they found that it really looked as though if you were on dupilumab, of course you're on dupilumab before you get sick, these were patients who were taking it for other reasons, those patients didn't die, didn't undergo ventilation. And there have been a few other cohorts that were looked at that supported this concept, that people who were on dupilumab seemed to be having less severe consequences. Of course, sorry for anybody, I should have clarified, dupilumab blocks the IL-4 receptor. So it's going to block IL-4 and IL-13 signaling. And is given for a variety of things, particularly things like atopic dermatitis. So those people appeared to be protected, or at least suggestive of being protected, which at least helped support our idea that there might be something quite important going on here. So what we obviously wanted to ask in the mouse model was why is IL-13 making disease worse? And so we looked at all our typical type 2 readouts to see what might be going on. And there was some reduction, but it was nothing very profound. I mean, realm alpha was a little bit reduced. There was very little, if any, impact on YM1. Goblet cells increased with infection, as you would expect for a type 2 response, but only sort of marginally decreased with the anti-IL-13. So there was nothing obvious in our normal arsenal of type 2 responses to explain why they were having more severe disease. So in Virginia, they did some transcriptional analysis to see what the differences were between the animals that had been treated with antibody to IL-13 versus the controls. And the most differentially regulated gene was a gene called hyaluronase synthase, which we here in Manchester, where we have the Matrix Center, and people like Tony Day who work on glycosaminoglycans, and Tara got very excited about it when she saw this data. And so hyaluronase synthase, as it says, makes hyaluronan. There are some other hyaluronan synthases which were actually also all regulated in this study, and we were able to show that in fact this synthase is downregulated when you block with IL-13. So hyaluronan is a non-sulfated glycosaminoglycan. It's throughout your body, and it comes in. This is a picture of it bound to a protein core, but these are these highly non-sulfated glycosaminoglycans, and they're in high and low molecular weight forms. They can be pro and anti-inflammatory. It's a whole area that I'm having to learn about. But importantly, other people had already shown, including Tracy Hussle here in Manchester, along with Tony Day, that hyaluronan drives lung pathology during influenza infection. And now a number of groups have shown that it's involved in COVID-19. That it is a molecule that makes COVID-19 worse. And we also saw one of the main receptors for hyaluronan is CD44, and we saw that that was also influenced by blocking IL-13. Tara just looked in the lungs of these mice that we got from the University of Virginia, and indeed they had lots of hyaluronan in the lung, and that was reduced when they had been treated with the antibody to IL-13. And really remarkably, once again, through Virginia, we got tissue from the university. Well, Tara got post-mortem tissue from COVID patients from Ben Bradley at the University of Washington. And it was clear that in the COVID-19 patients, there was much more likely to be hyaluronan in the lung of the patients who had died. And that it also in the serum, the levels of hyaluronan in the serum can correlate with immune status. So I think really strong evidence that hyaluronan is a bad player in COVID-19, and that itself is not novel. Other groups have shown that, as I said, but I think the real novelty is that this is connected to IL-13. I don't think anybody has ever shown that there's a relationship between hyaluronan and IL-13 to our knowledge. And so what Tara did to check that out is she delivered IL-13 directly into the lungs using this fusion protein that we use of IL-13. And sure enough, IL-13 alone delivered intranasally drives hyaluronan deposition and accumulation in the lung and along. So this is just throughout the lung, in the parenchyma, along the airways, and along the blood vessels, as well as increasing serum. So the next slide is my last slide before acknowledgements, and is that I'm increasingly captivated by the idea that IL-13 is a master regulator of the extracellular matrix for all sorts of interesting evolutionary reasons. But we've known for a very long time that it regulates regulates mucus, not just the amount of mucus you produce, but the nature of the mucins that you produce, the quality of that mucins, that's regulated by IL-13. We know from lots of work, but also some of the stuff I showed today on IL-13 inducing YM1, that it's regulating collagen cross-linking and deposition. Once again, there's lots of data out there to promote the idea that IL-13 regulates collagen from people like Tom Wynn and others. And now I think there's really strong evidence that IL-13 is also regulating glycosaminoglycans, a major component of the extracellular matrix. So, exactly why that is, I don't know. And these are such different components of matrix, and yet IL-13 appears to be regulating them all. And I find that personally quite fascinating. So I've tried to acknowledge the people who are involved in the COVID study, but just finally from my lab, the people who I have failed to acknowledge, I just wanted to acknowledge Brian, who maintains all our life cycles and keeps the lab running smoothly, and actually keeps us sane and happy and bonded even during the lockdown with things like weekly quiz nights. Stella does all our histology and is a really valuable technician in the lab. Some of these other people, I will mention their work when I talk about it in some future talk, and I've told you about Yasutas's work. So I think I've tried to acknowledge people as I go along. I'm sure I've missed people out, and who I have, I'm sorry, but thanks very much.
Thank you, Judy. That was fabulous. Thanks so much for sharing this very exquisite fine-tuning on how you can transition from the immune response to injury to the induction of tissue repair, and the fascinating roles and your perspective on the role of IL-13 on the matrix. That was really wonderful.
Thank you.
Okay, now before we finish, I would like to remind everybody that if you have questions, you can ask questions to Judy via Twitter. There's already a tweet that you can find in the Global IMMUNOTALK account that says, ask questions for Dr. Judith Allen here. So please find this tweet and reply to the tweet with your question. And do not forget to mention the hashtag Global Immuno and Judith Allen's Twitter account. Her Twitter handle is @Prof. Judith Allen. She will use that to answer questions. Judy, thank you so much again. I really enjoyed your talk, and I very much look forward to connecting with everybody next week when we'll have Sonia Sharma presenting her Global IMMUNOTALK. Thanks, everybody.
Thanks so much, Carla.
Bye, Judy. Bye-bye.
Bye.